Good morning, friends. It's a very special morning, I think, for me, and I'm very happy to be here today. And I want to thank Professor Yonath for taking the time from a very busy schedule to come here and share some thoughts with us. And I think that it is not every day that we are so privileged to have such an esteemed guest here in Mumbai. So I personally really want to thank you for that and for the effort, especially coming from the other side of Mumbai and having to go through all the traffic to come here to South Mumbai. And of course, I want to thank our friend Dr. Kulkarni and everyone in ORA for organizing this event in such a really so quickly, and everything was organized and people were invited. And thank you all for taking the time in the middle of a working day to come here and be with us. Just a few words. I think that Israel is very well known around the world for its scientific capabilities and achievements in recent years. Actually, I always say that, you know, in Israel we don't have any natural resources. The only thing we have is the human resource. And if the government will not put all its efforts to develop this human resource, so we are not left with too much. So I think that in Israel, since the establishment of the state, the government has been supporting the academic research. Maybe Professor Yonath, coming from the academic life, maybe she has a different point of view, but I think that basically it is something that is very much embedded, that we have to support the academia, we have to support the research. And from the other side, the academia cannot, or the research institutes cannot be somewhere that, you know, people sit here and discuss all kind of philosophical questions. They need to give also practical answers to problems that are more earthy problems. So I think this collaboration, at least from where I see it, has been working for many years. And Israel, I think, has the largest, the highest percentage of its GDP invested into research and into R&D, basically, which is almost 5%. This is the highest in the world today. I think that we can see, maybe I'm wrong, what we have about six scientists already that won the Nobel Prize. I think scientists, 5. Okay, 5. Yes. All right, for peace, exactly. 9 altogether. So 5 is still okay for such a small country, and I think that it really shows the level of the achievements of the Israeli science and technology. I really don't want to take too much of your time. You didn't come here to listen to me this morning. Please, Professor Yonath, we are all looking forward to hear your comments and your speech. Thank you so much, and good morning.
Morning. Thank you for coming, although I'm not an innovator, I'm just a scientist, basic scientist. But if I can just go back to the last sentence, that Orna just said, there are five scientists that got the Nobel Prize in Israel. During the last century, it means the investment that was done in the beginning of the country is now starting to give fruit. There is one writer and 3 peace Nobel Prizes too. But I want to quote in this regard. I want to quote the British ambassador that was introduced 3 days ago. That was introduced 3 days ago for in the occasion of establishing a UK-Israel Foundation for Science, for funding science. And he said that both Britain and Israel have, what I can say in my words, too many Nobel Prizes compared to the population. It means the ratio is very high. And I'm very proud of it, being Israeli. But as I told you, I'm a basic scientist, and all what I wanted to understand all my life is how nature, not only nature, also how man inventions function, but mainly how nature functions, what are the processes in nature. And I started as a basic scientist. I think that I'm until basic scientist, but on the way, at least there were at least two directions that we could become innovators even without thinking about it. And I think this is the beauty of basic science. It can become useful by the population immediately or 200 years later, like what happened to Mr. Newton when he was lying under the tree and an apple fall on his nose, and only in the 20th century missions to space started based on this falling on gravity. So let me tell you what we are doing. I assume that not all of you are scientists, so I have a very short introduction to why the process that I looked in was important. I took it from children's book, and I think children are the best to, if they understand, we should understand too, but they understand faster. Proteins are the workers in all the living cells that are doing almost everything we need. So I want to start with this protein, transporter protein. We all know its name, hemoglobin. He transports oxygen from the lungs to the muscle. Sensory proteins, clearly all of us see, speak, hear. Enzymes that you see here, this doesn't work, oh yeah, that chop our food. We are eating, and then the food has to be more and more reduced in size and in the way the components are being afterwards used. So enzymes are doing it. And the regulatory proteins, like insulin, are regulating the level of sugar in the blood and so on. I don't want to go too much. Maybe, I think in view of the investment of India in structural proteins, especially collagen, that was the main topic of the late G.N. Ramachandran. I think, in my opinion, one of your giants, or the Indian giants in science. So I also mentioned structural proteins. Proteins exist in every living cell. There are 25,000 different types of proteins in a human adult, and 100,000 billion cells in an adult, and each of these cells has lots of proteins. Even bacteria, the smallest cells, the smallest living organisms have proteins. Now, I want also to say that in nature, structure is function. It means these proteins cannot function when the structure is not correct, even if their composition is correct. So please, I'm now an enzyme, and my duty is to lift the glasses. And I do it with these two fingers. This is my task, my function. The same two fingers, if one of them grew there, won't be able to do the work, although it's exactly the same me and the same hand and the same fingers. So the fold or the positioning is very important. I have even a better example. See paper clips? So these are two paper clips in the right fold that can combine two pages. But I myself, with my own hands, took the same clips and just changed the fold to this one or to this one. It's the same paper clip, but it's not functional. So the structure is very important. Then we think that, for instance, enzymes, one of the models is that they have to work like lock and key. If this is the enzyme, it has an active side that should fit like a lock and key, and you can see it here, for instance. Now, it may look complicated, but if you focus for a minute, this is a protein that has to chop, to cut. And here, it does it here. All this is a protein, and it does it here. And just to show what happens here, I zoomed on it, looked at it from another direction. This is the substrate that it has to chop here. If the substrate doesn't sit exactly here, it won't be chopped, or it won't even go in. So it's very, very important that this nest here is ready for the substrate. there are 20 types of amino acids. Amino acids are the building block of the protein. So what you saw here is made of 20 types of amino acids. They all have the same backbone, but different nature. And the sequence of them, of the 20, for instance, the protein is made of 1, 3, 5, 17, 3, 9, whatever. So the sequence is determining the fold. This is built in the sequence. Proteins are very clever. And the sequence of the proteins, of the amino acid in the proteins, is determined by the genes. So actually, DNA is a string of instructions. Just string. DNA, this big thing that everybody talks about, it's a string of instructions. Many genes, and each gene is coding for one specific protein. So the sequence of the gene is determining the sequence of the amino acids, and the sequence of the amino acids determines the fold, and the fold allows the function. I just want to tell how important is this process. So the genes are in DNA, and this I think you all know. The language of the DNA are 4 letters, we call them bases, and they're making pairs, you see here. And these pairs, this language is now going through the DNA. It's not accessible because it is packed in a double helix, and all this is packed even more. So in this way, it cannot be translated, or it cannot be used. So first thing, the DNA is being transcribed to messenger RNA. The names are less important. Messenger RNA is like DNA, a little bit different. Its biggest property that we are using, its main property is that it can exist is a single strand, not a double strand, so it can be translated. The transcription from DNA to messenger RNA that I thought once that it's a simple thing, just copying, is a very complicated process, and Roger Korenberg got for this at the Nobel Prize in 2006. So it's, even this is really complicated. Now, from messenger RNA, we go to protein by translation. The RNA or the DNA, the nucleic acid language, are 4 letters. I already said earlier, 4 bases, 4 letters. Proteins have 20 letters, 20 amino acids. So the ribosome that does the translation reads in one language and produces in another one. Very clever machinery. So please forgive me again, but I really love this children's book. Here you can see the ribosome as the cell factory, and actually it's called cell factory. The instructions are coming linearly into it here and come out after they are being read inside there. You can think about any factory, car-making factory. The amino acids are brought on tracks, and all the tracks are very similar. The ribosome has to select the right ones. Here I show the amino acids as points in different colors, and they will be here inside, connected one at a time to the growing protein. So this is the newly born protein, which is a chain of those amino acids. And the decision which amino acid goes in is embedded in the code. The whole process is very efficient. I'm sure that most of you don't know what is GTP, but it's energy-rich molecules. And in my opinion, to add one amino acid to all this with just 2 of them, it's fantastically efficient, unbelievably, amazingly efficient. So there are a huge number of ribosomes that function in each living cell. Mammalian can have millions, 5 million in the liver, in one cell. Even bacteria can reach 100,000 in one cell. They act continuously, and they form over 15 bonds. Peptide bonds are the bonds between amino acids in 1 second. Now here I want to tell you about my experience. I was a very good student in chemistry, and I had to make one bond in my second year. This was an exercise. I needed 8 hours. For one bond, I needed 100 degrees centigrade and very low acidity. And the ribosomes are doing it in our body without warming it up 15 times second to 100 degrees, and they do it with hardly any mistake. 1 in a million. You can see here. The fidelity rate is 99.999. Just unbelievable for me. So what I said until now, DNA is a string of instructions called genes, and the language of these instructions contain 4 letters called bases. Each free of the bases is compatible for a specific amino acid. So the language is 4 letters, and each triplet is associated with amino acid. So what you see here is this is the ribosome. This is made of 2 parts. The messenger RNA with 4 letters, and I made every letter in a different color, so it's yellow, green, red, and purple. They are divided to triplets, and the first triplet to be decoded is in this particular case here. The ribosome binds the messenger RNA and also binds it so that the first to be translated is this one. Now you can imagine what would happen if the triplets would not be defined correctly. If the ribosome made a mistake and defined the triplets just one base to the right or to the left, then there will be a garbage protein. Just a nothing, because all the sequence will be wrong. Also, if it doesn't start in the right one. So there is a specific initiator to start, but it has to find it. So anyway, this part of the ribosome, the small part, the small subunit, and ribosomes are universal. They work the same way in all cells. First of all, the small subunit does the association, the decoding, and it has proofreading, really clever, the brain of the ribosome, and the large subunit is making the protein. So these are the working hands, and it will happen here. So I want to show you a movie, but can you close more light? I hope that you can see it. But the darker it's better. So the movie was made according to our result. There is not too much movie in here. Oh, good, thank you. By 2 students from the academy in Jerusalem. So look here, small subunit took the messenger. There are factors here that are not ribosomal, and they go out in a minute, the blue ones. The first track is called tRNA, is being brought with amino acid, and the large subunit comes and hugs the small one. Now we have an active ribosome. So the 2 subunits are separated until they come together. Now all the tRNAs are standing in line according to the messenger, according to the code, and come in and go out. The ribosome helps. And peptide bond, the connection is being made here. The protein goes through here and will go out. Now we took away the whole large subunit. We left only the tunnel for which the protein goes out. It's a very important tunnel. This is the way to protect the newly born protein. Like kangaroos have a pocket, this is even a better protection internally. So now you see the whole ribosome again. The protein comes from here. The messenger is being called read here. The tracks that are called tRNA are being brought with factors, cellular factors, and go out from this side. And it will all continue until there is a stop codon, and then the tRNA will be replaced by recycling or release factors. The 2 subunits can separate. Now they cannot be active. Protein goes to what it has to do, and the tracks too. So this is the story, and I want to jump, I think. Yeah, I want to just to show you how they are built inside. The small subunit is now rotating here, and you see it's made of many, many components. One of the components is RNA, and all these others, the RNA is in silver, and all the others are proteins, different type of proteins. There are 20, 21, depending on the bacteria. All what I'm showing you is bacteria. This is what we worked on, and until recently, the only structures known. And the large subunit, the same color code is running around. And whoever wants, there are here some statistics, but it's not important. What I want to show you, which is important, are the surfaces that will see each other when they come together. What you saw until now is 2 separate ones, and when they come together, they will meet this way. And the surfaces are RNA rich. almost only RNA. The bond will be made here, the chain grows here, the decoding is here, only RNA. So this shows that the ribosome is a machine that is made of RNA. It's an enzyme of RNA. Until now, I told you almost everything is done by proteins. Yeah, that's correct. Almost everything is made by proteins, but not the proteins themselves. For this, the cell has a different machine, and we think that we identified it even as a prebiotic machine in the modern ribosome. So it was RNA, there was RNA before proteins, and we think that we could identify the very, very, very ancient machinery that could make bonds in the RNA world, and it's still functioning in the ribosome today. It's billions of years. So this is one of the projects that is real basic science. I'm not sure that any innovation can come out of it, but I don't know. Okay, let's talk about more practical. You see here several geniuses, composers and writers that I'm sure you know Mozart. Kafka, Orwell, you know or you don't know, it's not so important. What is common to all of them, they all died before 1950. They all died very young. He's the oldest, 47 years old. Those didn't even reach 40. And they all died from infectious diseases. And why did I say 1950? Middle of last century? Because then antibiotic came into use. And before this, people died from very little normal infectious diseases like pneumonia. Mozart died from pneumonia. He had a destroyed liver, but the killer was pneumonia. So antibiotics, what are they? Oops, sorry. The natural antibiotics are the weapons that bacteria from one type use for interfering with cell life of a different type. So I'm sure that some of you, maybe all of you know, that in the social life of bacteria, there are wars. They are not all of them are peaceful. So if bacteria, one type of bacteria sees that another colony is trying to get there, they have weapons, they produce weapons. And we try to use these weapons that are called antibiotics for medical uses. And because of the fundamental role of the ribosome, many antibiotics target it. So now I hope that you agree with me that protein biosynthesis from the code to protein is very important for life. No proteins, no life. So if you kill, or if you not kill, if you stop the function of the main factory, in the generation or in the next generation, the bacteria will be killed. Now, generation bacteria is between 10 minutes to 6 hours. The pathogens are 4 to 6 hours. Therefore, we take antibiotics 3 or 4 times a day. And by a few days, there will not be any more functional ribosomes, and the bacteria will die. This is the idea, okay? Agree? About 40% of the clinically useful antibiotics target protein biosynthesis, and most by paralyzing ribosomes. And reading the Bible as a child, I thought that it's appropriate to compare between antibiotics and ribosomes, like between David and Goliath. So antibiotics, their weight in chemical units, which are not important now, is less than 1,000, 600 to 900. And the ribosomes in bacteria are 2.5 million. So you see, this very small thing has to paralyze this huge thing, like the stone that David had to paralyze Goliath. Now, Goliath was covered and protected well all over, except for hands and forehead. And David took the little stone to the forehead, because the forehead was the most important piece that was for life. Hitting here is whatever is behind it, whereas hitting the hands would not be so successful. So this is the way we like to compare, and I want to show you what happens. All known antibiotics that work on ribosomes target functional sites. So if you remember, this is the small subunit, and this is where the messenger, the code binds, and also the tracks, the tRNA. So they can sit there and hamper the function there. or in the active site where the bond is being made, or in the tunnel. Or if you remember in the movie, there were hands going in and out, helping. So this is one of the hands here. And this targets the screw of the hand, so it freezes the motion. So I want to show you in more detail just the large subunit with 3 different antibiotics inside it. I want to tell you that all what I'm showing you are experimental results. We put together in one picture 3 antibiotics. They are never together. Each of them is okay by itself. Chloramphenicol, it is sitting in the position of the first tRNA. Clindamycin is sitting exactly where the bond has to be made, and erythromycin in the tunnel. And this is a bonus in my work. I wanted to understand protein biosynthesis, and here I'm telling you about antibiotics that stop it. I never expected to be able to do this, to see 600 on 2.5 million. I just didn't believe that it's possible. And you see, we are there. We have now the positions and the modes of action of all antibiotics that they target ribosomes, except for one. It's 30 different ones. So if we look, if you want to see better, for instance, erythromycin, this red one here, think about a large subunit that is cut this way, in the middle. So what you see here, the beige is the ribosome. Here is the position where the peptide is being made, the protein is being made, and it would have gone out through here, through the tunnel, if erythromycin would not stop it. It's like a barrier. And if you want to see the barrier better, let's look at it from top. We look into the large subunit. This is the tunnel, and this is the way erythromycin blocks most of it. Not all of it, but not enough for a protein, the newly born protein, to go through. So I don't want to say much about chemistry, but this is the main structure of erythromycin. It was the first antibiotic against ribosome that was discovered and used in the middle of last century. And it was found to be very, very useful for many, many infectious diseases, but not so good in the stomach because it's not very stable in the stomach environment, which is very acidic. So companies try to improve it by changing here and here. And I want to show you the changes, more important than the equation. It's a view into the tunnel. Here is the tunnel. I enlarge it, and erythromycin binds this way. Very well, nice binding. Now the change, clarithromycin, one little addition here, which made it more stable and also better. It takes a little bit of the available volume. So it's a successful change. And I want to show something even more interesting, this one. So this is addition on this position, a longer hand. And I want to tell you the companies did it before we knew what the structure is. They didn't, they just tried many. They didn't select until the selection was done what does and what doesn't bind to the ribosome. So when we looked at this, we thought that we understand something that is unique, and it's a structural basis for dosing. Yeah, you surely know that the lower the dose, the better, because the metabolites of all drugs are poisons. So if one can prescribe 150 milligrams twice daily, that will have the same effect as 500 milligrams 4 times a day, it's very wanted, useful. And we, first time, I think, in the literature that somebody understands how this works. This is data that I got from a doctor. They just told me how they prescribe. And I think that I understand it by the stronger attraction here, that the drug stays in the ribosome longer and can be more useful. So this is one payoff, direct payoff. Now, I told you that ribosomes bind the antibiotics in the functional sites. All antibiotics target functional sites. And I also told you that all ribosomes work the same way, so the functional sites are hardly conserved. There are not very many differences between the functional site of a bacteria and of the patient. And I don't think that I have to tell you we want to kill the bacteria, not the patient. This is, we don't need a meeting for that. So how does it work? It works on the, only in, many antibiotics are not useful because they don't distinguish. But when there is distinction, they are. And the distinction is based on subtle differences, very small differences. Please have a look. I think you can see, yeah. This is the walls of the tunnel. Ribosome is all over here. These are many antibiotics from the same family as erythromycin. It's called macrolide family. So they have daughters, granddaughters, sons, and whatever you want, lots of new antibiotics, all bind in the tunnel, all block it better or less good, all interact with this here. And this here is one of the tunnel wall, and for those of you that are not chemists, the name iodine is not important. The number, running number, is just a running number of the base, but the chemistry is important. So it's A2058 in bacteria, including all bacteria, including all pathogenic bacteria. So this is the way the binding is. Sorry, I have to put some chemistry. The interactions are here, and they are very favorable. That's bacteria. You bacteria are the pathogens. Even archaea, which is a little bit higher in evolution, is already like human. Human, have a look here. That's the only difference between bacteria and you or me. That's it. Isn't it? It's also amazing. So, and when this is here, there is rejection because it's too close. And there is no binding. Ah, that's all. Isn't it fantastic? Yeah. Okay, so now I told you about the great things about antibiotics, and let's go to the bad things. There is a resistance, worldwide increasing resistance, and I hear here in India, everybody asks me about the super bug. It means resistance to all types of antibiotics. And this causes an urgent need for additional effective drugs. And if any of you think about innovation, there is a lot to do here, a lot to do. It's unbelievable. Companies are not very happy to produce new antibiotics. It's a lot of investment, and then antibiotics are not used for many years, like normal insulin or blood pressure pills that are taken by people over 50 with life expectation of 70, 80, or 90. It's a way to make much more money than 1 week or 2 weeks of infectious disease, and they cannot charge so much for them anyway. And they know, as we know, that resistance will always come. What we think is that we may be able to minimize resistance or make longer time until it arrives. But we are sure that we cannot, not with normal antibiotics, to eliminate resistance totally, because bacteria like to live. But let's talk a little bit more. I think that the fact that the companies don't want to hesitate to make new antibiotics or to invest in it is going to be very destructive for humans and also for the companies, because people will start to die young again, and they will not have patients to buy the money-making medicines. But anyway, how do antibiotics acquire resistance? It means how do the bacteria learn to acquire the resistance? Prominent mechanism is to modify the anchors that are bonded. And how do the companies try to partially combat resistance, or how do we have to, the ways to do it, partially because bacteria like to live. New compounds with additional anchors may compensate for the lost anchors that were modified for resistance. and also synergism, drugs that made from two components. Now, new compounds like the one that is successful is azithromycin that was discovered, I think, or made, I think, 15 years ago by adding just one atom to the ring. Instead of 14, they have 15. It's a fantastic idea. And the second is synergism. Find couples, pairs of compounds that can bind each of them, maybe not very strongly, but together they can enforce each other, block each other, glue each other. For instance, Synercid, it came to the use 10 years ago. It has two components, and again, the chemistry is less important. What you see here is the tunnel wall, one component, second component. They are like glue, and they don't let go out, and they made me a bit happier when I heard about it, from here to here. And indeed, this is what we are doing now. This is Synercid that I showed you, and this is a couple a pair that we are working on. It's called lancamycin, lancaciding, a funny name, lanka, because it was found, it is made by a bug that lives in Sri Lanka. And we found that they enhance each other. They are known since the 60s, but nobody actually used them the way we did. They are enhancing each other, and this is what we are trying now to improve. They are actually not yet ready. They were published, the publication about them came out 3 days ago. So I want to show you a little bit more about antibiotics, and then tell you about another innovation that came to us absolutely unprepared. If it agrees to come. Oh. So first, antibiotic is universal, not useful, but explain how it stops the interaction of the code with the ribosome. Just doesn't let it go. Freezes the motions and doesn't let it go. Sits on the path. So it's enough a little thing on the path that stops it. Second is tetracycline, maybe you know it. And it blocks, it occupies the same position as the second track should take. So first is there, but the second cannot come. So that's the end of the story. The third is what we talked earlier, erythromycin, that goes into the tunnel and shows that a little protein can be made, a little 5-6 bonds, but not more. They cannot continue through the tunnel. And the last is the one that sits exactly where the bond is. So here the bond will be made in the whole ribosome. Now we look at it and it sits here. So connection cannot be made, and that's it. So these are 4 examples to what we know today. And I want to tell you about the next innovation. So the technique that we are using is X-ray crystallography, and I will say 2 words about it, but I will not really explain it. It is needed when people want to see results or to understand processes at the atomic level, like I showed you now. Just one atom changes between bacteria and human. For this, you need a method that shows the atom. And neither of eyes or microscopes can do it. Therefore, we use diffraction, not even looking through, of X-rays. The method is one needs to have crystals. The crystals diffract the X-rays according to its structure. So please forget all the pink things. Just look at the crystal being here. Oh, the pink is now red. The crystal is here, and this is the diffraction. The beam comes from the side. Most of it goes through, but some diffracted. Long and short show if the diffraction is strong or weak, and the direction, and all directions. So we have to collect all directions. In each direction we see something like this. We have to merge them together. That's not very simple. There are methods that have to be used, and sometimes it works, sometimes innovation is needed here too. This is us, the crystallographers. You see this little thing? with computers, we can get at the end maps that are topological maps that show where the electrons are. You can see here. Here are the electrons. It means atoms. And you can accordingly build a model of your structure, of your compound, either small or even parts of ribosome or proteins. And this way you can get it. So what's clear, one needs crystals. One needs crystals. Crystal of sodium chloride is known to all of you. You buy salt that you put in the salad. It comes already crystalline. So is sugar. What is a crystal? It's an entity that has periodicity, full periodicity in 3 dimensions. This one, and that one. So salt likes to do it. This is salt. Sugar likes to do it. Some proteins could be crystallized even by a 6th grader, according to published procedures. But ribosomes, you remember them? So flexible, so heterogeneous in structure. There was a big question mark which was actually say no. Ribosomes cannot be periodically packed. There were papers about it, articles, and even books written by 20 groups, very good groups, that tried to crystallize ribosomes from the minute they were discovered until they failed. And the reasons are all what I said before, including, and in my opinion most important, tendency to deteriorate. Tendency to deteriorate, they deteriorate when they are prepared by human, they deteriorate in the test tube within 3 or 4 days. So why did I decide that I can do it? Or was I ready to try? Because I had an accident, I had time to read, and I read in journals about the bear's life. Bears in the North Pole, they go to sleep in the winter, about October, November, and get up April, May. So they are sleeping for 4 months, and people went in the 70s. Some delegations, mainly zoologists, I think. I don't really remember, and I couldn't find the paper that I read, the journal. But they say that while the bears are sleeping, the ribosomes are orderly packed in monolayers, like apples, a monolayer of apple, inside the membranes of their cells. So what did this indicate to me? That ribosomes can be orderly packed, in contrast to the publications. Every bear does it every winter. It means it's not just one lucky occasion. It's a process that is done. So if bears can do, maybe we too. But I thought, why should the bears do this? And in my opinion was that bears will get up at one point, the winter will be over, and then they have to do all types of things they didn't do while they were asleep. They mate, they look for food, they run, what bears are doing. For this they need proteins. For proteins they need ribosomes. And if they would not find a way to maintain a big storage of active ribosomes, they will be deteriorated by the end of the winter. So there is no reason to get up. So I thought that this is what we have to do, to crystallize ribosomes from bacteria that live under extreme conditions, assuming that this will be more stable, more robust. And we use the Dead Sea, hot springs, and bacteria that live in nuclear facilities, and look for procedures that extend the life a little bit. And I was lucky in the Weizmann, a procedure like this was published 20 years earlier. So the Dead Sea, just to show you, this is the sea, very small, very salty. This is salt that comes out. It's so concentrated. Although it's so concentrated, and although the name is dead, there are 2 types of bacteria and 1 algae that live there, and these are colonies of one of the bacteria called Haloarcula maris morti. Halo is salt, Arcula is a type of bacteria, maris is sea, and morti is death in Latin, so this is the name of this bacteria. It grows even on the salt, you see it better here. And we use this and the others, as I told you, and we have beautiful crystals. We have 25 types of crystals, and some of them are suitable for us and for others. Until then, from the time I started, I was called the crazy, the dreamer, the fantasionaire, the village fool. Some even called me the liar, but until they repeated it. But this was not the important thing. The important thing is that we got the crystals and we went to measure them. And we measure in synchrotrons. These are accelerators. This one is in Grenoble. Israel is a member of it as a European country, but now I heard that India is a member of it. India bought a beamline there. So this is where we measure. The idea is that there is acceleration, and there are, on the tangential, we make the people that build the synchrotron made experimental station like this one, and there are many of them, each of them with parameters that are suitable for the specific research that is made on it. And we went to measure in 1986, which means 6 years after we started, and we got fantastic diffraction. For those of you that know what diffraction, and for those that don't, the more data here, the more measurements that can be made here near the edge give the higher resolution. Here it's very low resolution, you can't see atoms, for instance. So this is what we got. It's wonderful. But after 0.1 of a second, this is what we got. And we needed, so we had only one view, and we have to go all around the crystal. So we got this. And this was, we repeated it many times in many synchrotrons, and it was the same. So this was like a dead end in our research. We felt like we climbed one Everest. We thought that this is the Everest. Then we discovered that there is a bigger one. We have to go down and up again. So I don't want to go into all details, but we found a way to maintain the life of the crystals for almost ever. This is the experiment in Stanford synchrotron. This is me. You see my face are not very believing. But we found a way by cooling. And why cooling? By flash cooling. Why flash cooling? Because I thought that there are two steps in the deterioration, in the damage of the crystal. All living tissues are destroyed by X-rays, by Röntgen rays. We know it even when we go to take a normal X-ray of the body. The people that work there are always protected, and our exposure is minimal. So the first destruction is physically controlled, and this is breaking of bonds. Now, if a bond is broken by the X-ray, there are now two parts of the bond. Each of them is unhappy. One is when the bond is made by electrons. One has too many electrons, one lacks electrons.